Okay, so the chloroplast has small double standard circular DNA molecules. Double standard circular DNA molecules. Circular DNA molecules and ribosomes, right? So it is circular. It is also circular in mitochondria, but it is single in mitochondria. Let me check it. It's kind of confusing, right? So possess single circular DNA molecule. Now extract given is like it's double standard circular DNA molecules in chloroplast. So double standard in chloroplast and ribosomes. Like now chlorophyll pigments are present in thylakoids. So what has thylakoids, right? It has chlorophyll pigments, right? Now ribosomes of chloroplast, these are smaller, that is 70 years than the cytoplasmic ribosomes. These are eukaryotic has 80 years. Now the ribosomes given by George Pellet in 1953. Now these are ribonucleic acid, RNA and proteins. So these are ribonucleoprotein, right? Now eukaryotic has 80 years, prokaryotic 70 years. Okay, so now each ribosome has larger and smaller subunits. So in 80 years, 60 years and 40 years, in 70 years, that is 50 years and 30 years. Now S stands for sedimentation coefficient. It is indirectly a measure of density and size. And so both 70s and 80s made up of two subunits, larger one and smaller one. See in the diagram the larger subunit how it looks, and small subunit can be asked in the exam. Okay, now cytoskeleton in eukaryotes, right? Now cytoskeleton made up of microtubules and microfilaments and intermediate filaments. These are proteinaceous structures and present in cytoplasm and function as support motility and maintenance of the shape of the cells, right? And this is it, like cilia and flagella, these are outgrowths of the cell membrane, and cilia help in movement of either the cell and the surrounding fluid. Flagella mainly longer and for the cell movement, right? And now the prokaryotic bacteria also possess flagella, but these are structurally different from that of the eukaryotic. So prokaryotic bacterial flagella and eukaryotic flagella, they are structurally different, right? Now there is core, that is axoneme, okay, and cilia flagella have the plasma membrane. Now they have centrally located microtubules appear, and 9 they also have 9 doublets of radially arranged peripheral microtubules. So this is why it is 9+2 arrangement, right? 9+2. These are for eukaryotic flagella, right? And now central tubules are connected by bridges, and central tubules are connected by bridges and enclosed by central sheath, okay, to one of the tubules of the each peripheral doublets by radial spoke. So 9 radial spokes are there, and now also the peripheral doublets interconnected by linkers, okay. Now both the cilia and flagella emerge from centriole like structure called the basal bodies. Now centrosome and centrioles. So centrosomes, centrosomes, these are the organelles, okay, usually containing like 2 cylinder structures. These are the centrioles, and they are lie perpendicular to each other. They show the cartwheel like appearance. They have 9 evenly spaced peripheral fibrils made of tubulin protein, and it is triplet. And the central part of the proximal region of the centriole is also proteinaceous, and this is hub. And this hub is connected with the tubules of peripheral with the peripheral triplets by the radial spokes. So they are also 9 in number. Now the centrioles form the basal body of cilia and flagella and the spindle fibers. So centrioles form the basal body of cilia, flagella, and also centriole also form the spindle fibers that give rise to spindle apparatus during the cell division in animal cells. Now going to the nucleus. Now nucleus is discovered by Robert Brown. Very important line. and it is first described by Robert Brown. It's given even in the beginning as well in 1831, and later it is stained by the basic dyes, and the material was found inside the nucleus was given the name like chromatin by the Fleming, right? The Fleming, right? Now you see there is nucleus. Now what does nucleus has? So nucleus has like the nuclear envelope. It is double layered, and it has nuclear pore, and then inside it has nucleoli. It is nucleolus. There is active ribosomal RNA synthesis, and it is non-membrane. It is then